Hello guys, today I will discuss the proteins involved in eukaryotic DNA replications. Previous video I will also discuss the protein involved in prokaryotic DNA replications, and in this video I will discuss in eukaryotes the replications. As you know that in the replications, three phase participate. First, initiation. Initiation means all the proteins and factors are ready to perform their functions. Elongation, here each proteins are ready, will be ready to perform their functions. And terminations, the products will be found and every proteins will dissociate from the DNA. So let's further discuss the initiation. In initiation of the DNA replication in prokaryotes, MCM, mini chromosome maintenance, is act as helicase like in prokaryotes. Hexameric protein, DNA in prokaryotes, DNAP act as helicase, and here MCM act as helicase like proteins. RPA, replicative protein A, it also act as the single stranded binding proteins in eukaryotes, and in prokaryotes SSB proteins participates. CDC 45 act as recruitment of DNA polymerase alpha. Next, elongation, three proteins participates. DNA polymerase alpha help in initiation phase, DNA polymerase delta help in elongation phase, lysing phase, DNA polymerase epsilon help in leading phase. Everybody know that what is the lysing and leading. For example, just like DNA fork, leading, the nucleotide elongate continuously, but in lysing, nucleotide elongate untrapped, dissociated form, like as. But in leading, the nucleotide elongation will continuously. He is the lysing strand, he is the leading strand. He is the lysing strand, he is the leading strand. Here nucleotide elongation untrapped, here continuous. PCNA act as sliding clamp, but in prokaryotes beta clamp act as sliding clamp. FEN1 act as endonucleases, primases also involved. I know you you all know that what is the primase. Primer is the RNA molecules initiate the elongation of the new strand. RNase is the exonuclease 5 prime to 3 prime nucleus topoisomerase. Topoisomerase is a exonuclease type endonuclease type of the enzyme, because these enzymes remove the tension stress of the DNA. For example, if two strands of the DNA very compactly arranged to each other, between two strands a gap can say partial gap found, and this gap does not initiate the transcription or replication of that type of the DNA, then we could use the topoisomerase enzyme. This enzyme bind to the double stranded single stranded of the DNA and remove these tensions, and this DNA will be relaxed after adding the topoisomerase enzyme, and we can easily transcribe this DNA or translate replicate this DNA. This is two type. Class 1, remove supercoiling DNA, just I says break is break only single stranded DNA, not double stranded DNA. Like only this DNA break single stranded, not both stranded. ATP independent, it does not require any ATP during the break of the DNA. Cut and ligate is also helping the cut and ligation joining of this DNA. Nick also created. So guys, I will also discuss Nick and gap. What is the Nick and gap? Like is the DNA base, nitrogenous base also here A T T T G C T A A A C G. If one base here will be removed here from this DNA, one base will be removed. This is called the nick. But if back bond like in nucleotide, back bond sugar this is the back bond removed from this DNA, like sugar, phosphates and nitrogenous base, then it is called the gap nick. This is difference between them. Class 2 topoisomerase break double stranded DNA is the break both stranded of the DNA together. ATP dependent is required the ATP during the breaking of the DNA. DNA garage is the subclass of the class 2 topoisomerase, remove tensions of the DNA. So I hope you guys understand very well all these proteins that involve in the DNA replications. Thank you.